Okay, we're going to continue with proteins, talking about isoelectric focusing, and then in the next videos, we're going to go over more protein analysis techniques and just some separation techniques in general. But starting with isoelectric focusing, which has to do with the protonation and deprotonation states, isoelectric point, pKa, and pH, all that stuff that we talked about last time. I don't remember if we did this last time, but isoelectric point, what is that? Isoelectric point is when the pKa is equal to the pH. Okay, so one thing that we need to know about that, all right, we can change the pH to whatever we want our environment to, okay? So the PI, the isoelectric point, also known as the PI, is going to be based on the pKa. So whatever our pKa is equal to, our isoelectric point will be equal to, okay? And if we don't know the pKa of something, or the isoelectric point of something, we can measure that using isoelectric focusing, okay? So the way we do it, and on the right side here, we have this kind of separation gel here. We have a cathode and an anode, and let's just say this is at like 0 minutes, because this is the same thing, just different timestamps. And we'll say this is at like 1 hour or something. Okay, so just to be clear on that, all right, and they put 3 different proteins in there, protein A, B, and C, and when you put them all in there, you're just putting them all in the same point. You just inject them in there, and they're just a blob of proteins, okay? Their protonation states are going to be all over the place. Some are protonated, some are deprotonated, and what these proteins are naturally going to do, they're going to slowly move, they're going to slowly migrate towards their isoelectric points, okay? So on this y-axis here, we have the pH gradients, okay? And the red one, it starts in the middle, and it migrates up to about 9, okay? So in this case, for the red one, we know since the PI is at 9, okay, and the pKa, once again, that's our innate property of the protein. That's not going to change. We know that our PI is equal to just above 9. I'm just going to round it to 9. So our pKa is 9 for A. Okay. For B, this is very, very simple. For B, it's at about, we'll say 5.5. B equals 5.5 for pKa. Okay, and for C, it's at about 4 for the pKa. All right, so naturally, these are going to want to migrate, okay, and the way that that works is because the cathode and the anode is positively charged, and the cathode is negatively charged, okay? So that means anything that has a positive charge is going to migrate towards the cathode, and you can kind of see it represented. They have the red one as positive. Okay, they have the red one as positive, so that's going to migrate to the cathode. That makes sense. Okay, and the yellow and the green, which are labeled as negative, are going to move to the anode, which is positive. Okay, and that can be confirmed with our knowledge of protonation states. Okay, we now know the pKa's. We now know the pKa's of these. Okay, and when we're starting out, the red one, for example, the pKa of 9 is greater than the pH of about 6.5 there, okay? And just as a reminder, when the pKa is greater than the pH, our protein or our molecule is going to be positively charged. It's going to be protonated, okay? So that confirmation makes sense, okay? Next page. So we're going to go over, this is going to take like 2 seconds. Gel electrophoresis. I just want to make this clear on how it works. Okay, so very similar to isoelectric focusing, and you guys have probably done this before in class, but in case anyone needs a reminder here. case anyone needs a reminder here, okay? Just like isoelectric focusing, we have the cathode top, which is negative, and the anode at the bottom, which is positive, okay? So we're going to feed whatever, proteins or molecules up top, and over time, they're going to migrate to, wherever they end up, okay? They're going to end up someplace. If this is a native gel electrophoresis, native meaning original, like we don't change anything, it's going to take into account the charge, okay? So whatever is more negatively charged is going to have a stronger attraction to the anode, okay? And one thing we also have to consider is the size, which, if you looked at a gel in person, it's not obvious to the naked eye, but the gels are cross-linked. And what does that mean? Is they're kind of like a mesh. They're kind of like a mesh or like a spider web, okay, that gets more dense as you go down. Okay, so size is a big thing. They essentially get filtered where only the smallest ones can fit through the gaps of this mesh, of the cross-linked mesh, okay? And the bigger ones will get stuck up top, okay? So it's literally a filtering mechanism. So those are two big things. First off, a filtering mechanism based off size due to the cross-linked mesh or like spider web or however you want to think about it as. And the other thing is the charge, okay? The stronger the attraction to the anode, and at the same time it's getting repelled by the cathode, the farther and faster it's going to go. Okay, so that's gel electrophoresis. Just generally, that's how it works. And one kind of small thing that is low-key that we might need to know. This is an example of an electrolytic cell. Okay, we're going to go over electrochemical cells in the future, but just a small thing. This is technically an electrolytic cell. We don't really need to know that, but just a little Easter egg, okay? And blotting. What the heck is blotting? Blotting is, well, we have this gel, okay? It's not very, functional for data purposes just to have the gel with us. So we want to transfer it to essentially a piece of paper or fabric, okay? So that fabric is going to stamp. It's going to stamp the top of that gel, and it's going to essentially take a snapshot of those of the locations of the proteins that we just measured. So that's all that blotting is. It's just a snapshot, essentially, of the gel. A lot of people get confused because there are a bunch of different types of blotting that we need to know. We need to know southern blotting, northern blotting, western blotting. What the heck is all of those? What the heck are all of those? Excuse me. I'm going to introduce a mnemonic that makes blotting very, very simple. You may have heard about it before. It is one of the most useful mnemonics you will have on this entire exam. Okay, snowdrop. And all you need to know for blotting is that southern is used for DNA. Southern blotting is used for DNA. Northern is used for RNA. These O's are just placeholders. They mean nothing. And western blot is for protein. Okay? So that's all you need to know. If we're measuring DNA, we use a southern. If we're using RNA, we use a northern. If we're using protein, we use a western. That's it. Snowdrop.